So, yes, welcome everyone, each and everyone who have joined this session on advanced biotechnology and agrochem. If we talk about the biotechnology and its background, a confluence of advances has been taken care of in biological sciences and accelerating the development of computing, automations, and now these all innovations are coming into a big revolution, which is known as bio-revolutions. And now the days are not far that these revolutions can kick the agriculture and health and all the economies to have a significant impact. There are major new biological applications are actually already improving our response on global challenges, including the climate change, pandemic, and many things. If we talk about seriously the pandemic that is going on, the speed which scientists are actually sequencing the virus genome and everything, that's biotechnology. And these biotechnologies are actually rapidly introducing new technologies like CRISPR-Cas, we call it snipper technologies. And these all technologies are now trending a lot. RNAi and silencing, gene knock-in, knockout. These technologies are recently required by the economy with that full potential and attaining that particular progress towards this, target towards economy, societies, and our lives, what we eat, what we wear, what medicines we take, and how we construct our physical world. With those background, I'm very much happy to invite each and everyone, all the viewers on our CI Hive platform and on the Zoom, all the panelists, I welcome you to the session on advanced biotechnology. The context behind this session is we have a session chair, Professor Ramanathan from Rallis, India, who will be chairing the complete session. And today, the day two of the session, day two of the Agri the conference, that is Fifth Global Dialogue. We have started off with our pitch competition. So in morning we had a pitch competition. After that we had a mechanization and automation session. We have a post-harvest session. So this is one of the last session from Indian Standard Time, and we'll be covering the advancements in technology, biotechnology majorly speaking, and how this bio-revolutions can take up. So without wasting any time, I'm very, very warmly welcome Ms. Karen from Israel, Mr. Nimrod from Biofeed Israel, Mr. Raman Babu from Corteva Agri Science, and Professor Rajam from Delhi University, a renowned scientist from India. Over to Ramanathan, sir. Thank you so much, everyone, each and everyone, for accepting our invitation, and we are glad to have you as one of the participants and chairing the session. Thank you so much, sir. Over to you, Ramanathan, sir. Please go ahead.
Thank you, Shivam. Good evening, good afternoon, good morning, everybody, because I understand we are having a global audience today. I would like to provide a very brief perspective on what exactly triggered this kind of an event. So we live in a world that epitomizes the term VUCA world. In these times, we are staring at a future with reduced resources for agriculture that has to feed an ever-increasing population in an environment of unpredictable climate. In recent times, the researchers have realized that the time available to develop suitable solutions to keep up agricultural productivity is shortening continuously. It requires an integrated approach to create products, processes, technologies, and solutions at a rapid pace to meet uncertain demands under uncertain conditions. It's a monumental task. Just see the kind of challenges India has been seeing in recent times. Monsoon playing truant or dumping a deluge at the wrong times, the fall armyworm menace in maize, the pink bollworm outbreak in cotton, sucking pests in cotton and a variety of other crops. To top this off, the decisions of policymakers and regulators impacting farmers' crop choices and the access to various key technologies, they make agriculture a daunting realm. While it is important to accelerate solution development, it is equally important to engage with policymakers and regulators to ensure that appropriate solutions are developed and, more importantly, deployed. And these solutions have to meet the metrics of environmental and business sustainability. All of these are key focus areas. So today we will probably delve a little deeper into solutions that are coming from chemistry, biology, biotechnology, and more recently, ideas coming out of artificial intelligence and much more. And I will very briefly introduce the distinguished panel we have today. First, Dr. Nimrod Israeli. He is an agri-industry keynote speaker, writer, and opinion leader. He is the CEO of BioFeed. But just going back a little bit, he has a PhD in ecological entomology from Hebrew University in Israel. And like I was actually chatting with him a little while ago, he was doing his master's degree when I was doing my postdoc fellowship at the Weizmann Institute right across the road. He is also the founder of the Green Valley Initiative. And Green Valley is working hand in hand with farmers to help them increase the quality and quantity of their yield to export and increase their income. BioFeed developed novel non-spraying crop protection technologies and protocols, which we will hear from Nimrod, hopefully, which Green Valley is using with its farmers. He spends much of the time in Asia and Africa, working with farmers, stakeholders, policymakers, and governments to form a better future for the agri industry. He serves as the director of the Africa Organization of Technology in Agriculture Conference, IOTA, and Expo 22-23. Then actually, Professor Rajam, he is a professor who teaches and conducts research at the University of Delhi South Campus in the Department of Genetics. His research focuses on plant genetic engineering and RNAi, on the development of crop plants for disease and pest resistance, as well as improvement of other agronomic traits using RNA and microRNA interference strategies. He is a fellow of many reputed science academies in India and also is an advisor to various bodies. He is on the editorial board of a few peer-reviewed journals and serves as a referee in many others. He has published more than 25 scientific papers in peer-reviewed journals in just the past 5 years. He was a co-editor of the 2-volume book entitled Plant Biology and Biotechnology, published by Springer. He has guided 36 students to their PhD degrees and has more in the pipeline. He has received extensive funding from government agencies to carry out research in key areas to address biotic and abiotic stresses of agricultural interest. Then Ms. Karen Moshelian. She is CEO at Biotech, an innovative company that helps optimize plant growth in changing climate and different soil water conditions by effectively screening and selecting the best performing plants and chemicals. Works with reputed scientific institutions like Wageningen University, Mars Wrigley, ICL, Haifa Chemicals, Göttingen University, Florida University, Tel Aviv University, and many more. After finishing her bachelor's degree in business management and economics from Technion, she did her MBA from Tel Aviv University. She has extensive experience in finance, management, operations, and strategy. And last but not the least, Dr. Raman Babu. He is the digital ag R&D lead of Corteva Agri Sciences for the APAC region. He is an ardent advocate of accelerated genetic gains through technology-infused modern breeding programs. In a career spanning over 20 years, he has developed genomic products and strategies that impacted the breeding pipeline across crops in national, international, and corporate organizations. He has worked both in the public sector as well as the private sector. He established trusted partnerships with many research institutional partners such as Embrapa in Brazil, ARC in South Africa, PARI in Kenya, ZARI in Zambia, ICAR in India, NMRP in Nepal, NMRA in Vietnam, and so on. And they are instrumental in deploying value-added corn hybrids across Latin America, Africa, and Asia. Babu has published more than 60 research papers in peer-reviewed international journals. With that, I invite Dr. Nimrod to start his presentation and share his thoughts with us. Dr. Nimrod, over to you.
Thank you very much for the presentation, and this sexy voice is not with me all the time. It is now because of a flu, and I'm glad that I can speak, because 2 days ago I couldn't speak at all. So apologies. And I will share with you two stories. I hope it will give you some perspective. The first one is about technology and about how much technology can change our lives and how beautiful it is. I grew up as a farmer. My father was a farmer. And when I started working as an older person after the Israeli service in the army, sorry, I was doing crop protection. I was spraying all the time. And at that time, I started to understand a couple of things. One, sprays are not a solution. They brought many problems with them, and they didn't solve many of the issues I was dealing with. Second, if I want to sell added value produce, I have a problem when I spray. And this was 30 years ago. To increase the value of my crop, I got into 15 years in the academy, and then I established Biofeed to develop technology that was still not available to challenge the number one pest that I was dealing with, which is a fruit fly. In India, you have other fruit flies. All in all, fruit flies are the number one group of pests for fruits and vegetables. And happily, I can say, and I will jump over many, many years and challenges. In 2015, my company Biofeed, we won a competition called Grand Challenges Israel, and we got to India. And we started working in India to develop a solution for the number one problem of the Indian farmers, fruit growers. If you want to guess, go ahead. Number one. So the number one crop is obviously mango. And the number one problem, pest of mango, is obviously fruit flies. And we are talking not about just one species, but about a few. And as you know, today, the average infestation is about 50%, but very often it can go up to 80 and even 100%. In particularly when it is rainy, there is nothing, simply nothing. So farmers must harvest the fruits when they are still green, small, the taste is not so good. And then when they market it, sell it many times, still about 50% of the marketable produce is infested. Under those conditions, to export is almost out of question. And this is a catastrophe for a country that is growing over 40% of the mangoes of the world. So we got the Grand Challenges Israel, and I got to India, and we did what nobody believed. Nobody believed it is possible to control those flies without sprays at all. Nobody believed it is possible to do it at a quality of export. And nobody believed it is possible during the rainy season. We did all that. By the way, nobody believed it is possible. We had a very short time of 1 year. And within 1 year, we developed a solution for the mango growers of India. We took the infestation from the average 50% to less than 1%. All of this with zero chemicals, zero sprays. This is like a dream come true, including during the rainy season. The result was so amazing, I couldn't believe it. I had to go and see in the field by myself just to be sure that these are really the numbers. And a short while after, the farmers, the Indian farmers which we worked with, asked me or asked the Minister of India to meet me. And I was lucky. I met Modi, Narendra Modi, and Prime Minister of Israel in the first meeting. That was very exciting. And then again, 6 months after, again in India. After that, I continue to see, okay, so how do we make it into action, into business? And here I found a few things that it is good to discuss. Even if, and when you have the dream product of the tomorrow, zero chemicals, no need for tractors, no need for sprayers, because the way it is working, I didn't say it is working, you just put it a post on a tree, just a few units per acre, about 4 units per acre, something like this. And you just put it on a tree, it attracts the flies by smell. And then those flies that are coming, they stand on this post, they lick something, they fly away and they die. So there is zero contact with the environment. And the first thing that I noticed is that the regulation, even for such solutions, which are obviously eco-friendly, are very, very difficult in India. And this is the first reason why today, from 2016 or 17, I should say, 4 years after, Indian farmers cannot have access to this technology. Africans do, Israelis do, but not Indians, where we started. So this is really, it's a shame. It's about regulation. And regulation shouldn't be like this for anything which is not coming in contact with the environment. And even if you touch it or eat it, there is no hazard. So this is one thing. And now I want to take you to a second story. And the second story is about the realization of the limits of technology. And just like there is a limit to power, there is a limit to money, to what money can do, I want to disappoint you. And I will do it from the place as the CEO of a company that is very technological one. I am very much like this. And because of this, I can say, we emphasize how much technology can give us solutions. And we emphasize we need more technology, we need more money. But wait, ask yourself, if it is technology, if it is money, how come that my parents in Israel, more than 70 years ago, when they arrived to a hill with only rocks and no water, no trees, just a few years after, they were already successful farmers. And this is not the case. And of course, technology at those times were not the same as it is today, and they didn't have the funds. My mother was a Holocaust survivor. She came out, she ran away from Europe with nothing, not even a family, no one. So how come that today, with much more funds, technology 1,000 times better, we see farmers, smallholders in such big problems? And here I want to say that I believe the future is not only technology. The reason we see farmers, smallholders, mainly in India and in emerging economies, struggle so hard is not because the lack of technology. Let me say it again, it's not because technology. It's even not because of funds. It is because of something that we neglected. And that is called business model. And before I ask if any of you know Professor Clayton Christensen, and what he said about disruptive innovation, that it's not about the technology, it's about the business model. When I understood this, and it was a few years ago, I started to study much better why some places are working better than others. And I found that it is definitely the business models that we introduce today to Indian smallholders are coming from the West. They are good for Western and professional farmers. They are not suitable for smallholders. Smallholders deserve and need a different business model. And this business model is what exactly Green Valley developed. And we are doing it when we understand much better their needs. And this way, only now, a week ago, I was notified that in 1 year, while we're still doing the pilot in Senegal, Africa, West Africa, we managed to double the export of mangoes from Senegal. This is unbelievable. This is only a pilot, double. Now, this is possible also in India. We can more than double. I can tell you that each of those farmers that were working with Green Valley, and what Green Valley, by the way, is doing, it is helping them to increase the quality and the quantity hand in hand. We work together. We don't sell them, we work together. When they do better, we do better. And then we take the produce all the way to the market, to premium markets, not to low quality markets. So they more than doubled their income in 1 year. And the country, Senegal, doubled their export. Of course, they get euros, they get dollars. This is the way forward. And with this message to look on business model, this is exactly what before you were talking about the conference in Africa. From now on, I believe that any conference that is talking about agriculture and technology should not ignore the importance of business model. I invite you to contact me and to follow me to talk about Green Valley. If you want to improve the income of the farmers and the economy of the country, it is coming together with a business model that is suitable. So this is my message to you. Technology, the sky's the limit, and we can do a beautiful world without sprays. And income and poverty, yes, we can do it. We can more than double the income of farmers in a very short time. Thank you very much.
Thank you. Thanks a lot, Dr. Nimrod, for sharing those two stories with passion. I invite Dr. Rajam now.
Yeah, thank you. Let me share my PPT.
So, Dr. Rajam, you will have 10 minutes. Dr. Rajam, we can see your screen, but we are not able to hear you.
Sir, in that case, can we go forward with next one?
Sure. And in the background, Shivam, if you can coordinate with Dr. Rajam, it will be useful.
Yes, please go ahead.
Sure.
He has got
Sure.
disconnected.
Yeah. Then I invite Ms. Karen to share her thoughts.
Thank you. Thank you very much. I'll be happy to. And if it's okay with you, I will also try to share my screen and talk to you about a technology which we developed in Israel. Let me know, by the way, if you can see my screen. if it's okay.
A moment ago, we were actually seeing this. Yeah, we are able to see the screen now.
Okay, thank you. Oops, it went away. Sorry about that.
It disappeared again. Yeah.
Yeah, sometimes it happens. Okay, hopefully this is okay now.
Right, we are able to see. Yeah.
So we are a company based in Israel, and we developed a technology for the scientific community, for the biotechnology and agrochemical market, or even bioag market. So everybody who has a certain business with plants and research plants, if it's in a university or it's in his company's R&D. And we developed, Israel is a very dry country, and we have limited water resources. So we deal a lot in Israel with the technologies that relate to water management and drought. Actually, India suffers from the same topics in a lot of areas of drought and also salinity issues in the soil. So these are environmental issues. They come because of the general trend of more cities and more people, less agriculture quality lands, but also because of climate change and extreme weathers. And we have to provide better plants, better resistant plants for tolerance with drought and salinity, or even different nutrients that will help the plants to grow in harsh conditions. So in Israel, we developed a system which was actually developed in the Hebrew University, two professors that were trying to come with solutions for these problems. And they used it for their own research, and then the university saw that there is also a broader need, and they actually commercialized the product. So I myself am leading the company now for 5 years, and I actually took it to the commercial phase. We are selling the product, which I'll explain more about it, already worldwide in Europe, North America, China, Israel, even now South Africa and South America. Hopefully very soon also in India, because I definitely see that India has the need and definitely can enjoy from this technology to accelerate the research in order to bring faster new products into the market that will help the farmers in India. This is really what we're doing. We're helping to accelerate the R&D processes, accelerate the research and the quality of research in order to reach to market faster. And we can save seasons and years of development. And I will show you a little bit more about how we do it. We have a lot of academic articles published by our customers, our users, about 40 till today. And you can see some of our customers here below. There are universities. Wageningen University is the #1 university leading in agriculture in the world. But we have many other universities, respectful universities, as well as also companies from the chemical industry, from the food industry, and also from biostimulants industry, and so forth. So let me just proceed. Okay, so what are we trying to solve? What challenge actually? So today it takes a lot of time in all of these genetics processes and also chemical processes. Where it gets stuck is really in the phenotyping stage, meaning at the phase where you have to check what it does to the plant, how the plant reacts when it comes in interaction with the environment. This is where a lot of time takes to really learn the effect on the plant. It takes at least 2 to 4 years, and sometimes even when you talk about environmental challenges, it takes even up to 15 years. It all depends on what you're trying to achieve. So it's like a medicine. Yeah, it takes very long time. And part of the problem is because it's people are looking with the eyes, don't have an objective assessment of the plants. They do it one plant at a time with no real good digital solutions. The profiling of the plant is being done only in a very small part of the day. So when you want to learn the effect of environmental conditions, it's very hard to learn about it this way because the environment changes even during the day. Sometimes it starts sunny and then changes to cloudy. And if you don't measure the plant all day long, you lose a lot of information about what is actually happening to the plant. So as you can understand, we study the plant. We study the plant response in order to provide answers to these challenges. One of the more complex issues to understand the plasticity of response of the plant is the environmental changes. And also you want to do it in a non-destructive way. You want to imitate what is actually happening in the field as much as you can. So we developed a system that is doing three main steps. One is applying different stresses. So automatically apply different irrigation regimes, different fertigation regimes, nutrients, salinity, whatever you want to measure. So it's automatically doing a certain differentiated treatment to each of the different plants in the array of plants and manage it automatically. And at the same time, measure the physiological response by the plant as well as the environment and doing the analysis for you. So you have like an analytical tool, software, and also the hardware to actually do the experiment. This whole process, instead of going to the field for a full season, takes only between 2 to 4 or 5 weeks. You don't need to go up to a full yield process. You already get a very good insight just by looking at the physiological response of the plant already in the vegetative stage. And of course, you get an objective assessment of your plants, which is very important. So, and you measure all the plants at the same time, 24/7. So all along the day, all the plants at once. So you really do a comparative analysis and you can really take, conclude and come to good conclusions based on your quick study with the system. So this whole process, we actually skip a lot of the trial and error that today is being done in the field. You do it in a greenhouse, and then directly after you do all these tests in a greenhouse, you go to the field with much better designed experiments. So you save all this trial and error, and you go already when you know exactly what to expect in the field. We have very high correlation between what we measure on the system and what is actually the outcome in the field. So you save time, you increase quality, and you save this very lengthy process of several seasons and years until going to the field. So you just have to do very few field trials in order to confirm your product and get faster to market, or your research, of course. Just to give you a quick example how it works. So plants are being grown on the table. You can see data being gathered at the same time. So it's in real time. You can actually learn about the plant response as they grow. This is just a 1 month trial, which you already have drought experiment with it. So drought and then recovery. And you can see that it works with many types of plants, as you can see here also in this picture. What we measure is a lot of different parameters from the plant. You see all these green parameters. We learn about the transpiration of the plants, the biomass gain by the plant, the water use efficiency by the plant. Stomatal conductance is a very important physiology parameter that is indicating actually the photosynthesis activity. So we have all of these different and even root activity. So we really learn about the plant productivity and growth together with the environment. We sense the environment at the same time, so we can learn about the interaction between them. And as I said, we also apply the different terms for the plant, so you can actually measure the response. As I said, this system applies for a lot of different, many type of plants. No, we didn't have any hurdles so far, from even small plants to big plants like trees. This is the system at Wageningen, for example. You can see they are doing drought and salinity stress tests over there. And what I'll show you here is today, most of the technologies, advanced technologies for high throughput phenotyping are imaging. We do it differently. We don't do imaging. And the thing is that if you see imaging, it goes, and this is the camera on top, it goes very slowly. You see it moving very, very slowly, only measuring few plants at a time. Look at the system below. This is our system, the plant array system. You can see all these green lights is actually measuring the plants all the time. We're measuring in very high resolution. So we can really learn about the differences and the reactions, the fast reaction by the plant. Sorry, something got stuck. Apologize. There is some background noise.
Yeah, Dr. Shivam, there is some background noise.
Until I fix it, it will be good to arrange it. Okay. So I'm trying to go forward. All right. So we are very unique with the system, trying to provide the answer for the physiological part, the activity by the plant, not just how it looks, but really about its activity, the productivity and the growth of the plant. It's kind of like the Apple Watch, what it's doing, learning the physiological status of the humankind, but of the man who is wearing it, but we are doing the same for the plants and many plants at once. So we really solved all the challenges I spoke about phenotyping for plants. We shortened the time, made it all automatic, so everything is objective. Many plants are learned simultaneously, so we really can understand very little changes by the plants. And we can really do very sensitive drought, salinity, nutrients, and additional tests to learn the plants fast and really to get to market within very short time. You can see the use of the system for genetic variety screening, water use efficiency, nutrients in bioag, seed treatments, physiology studying, early detection of biotic stresses because we learn the plant reaction very fast, and yield optimization of different kind of plants for protocols built up for irrigation or any other. So these are my contacts as well. So you can also contact me directly and I'll be happy to bring back to you for the continuance of the session.
Thanks a lot, Karen. Now I invite Dr. Babu, because I don't think we still have Dr. Rajam online. So in the interest of time, I request Dr. Babu to go ahead.
Yeah, Ram, can you see my screen and can you hear me well?
Yeah, to both it is yes. The only thing is the presentation is not in the slideshow mode. Yeah, okay. Now it is. Yeah, please go ahead, Bob.
Yeah, thank you very much, Ram, for the kind words of introduction, and thanks to CIA and the organizers for giving Corteva an opportunity to share our perspective on ag biotech. I will take the next 5 to 6 minutes just to highlight one particular aspect, one particular function of ag biotech that I personally feel is relatively little known as compared to some of its cousins, like, be it genetic engineering or a more fashionable recent one, genome editing. So as compared to this, the whole genome prediction or the genomics assisted breeding aspect of ag biotech is relatively little unknown, and I wanted to just spend some time on how this is impacting plant breeding, the seed product development in a big way, and it's happening across the globe. Just wanted to shed some light on this one. So just to provide, considering the heterogeneity of the audience, just wanted to give some context. Say, for instance, the ag biotech is very wide, and it can have pronounced impact on seed sciences in terms of developing seed products, and also on crop protection science, developing crop protection products. So today we are not discussing on the crop protection side. I am only talking about the seed science. And again, within seed science, the ag biotech can have, again, there are at least 3 well-known sub-functions within ag biotech. So genetic engineering, where you go across taxonomic boundaries. Then you have genome editing, where you can insert, delete, or substitute a particular base in the DNA. And then you have this molecular breeding approaches, wherein it's very similar to plant breeding. It is only the difference is in plant breeding, the selections used to be based on phenotypes, based on the measured traits, whereas in molecular breeding, the selections are assisted by genotypes as well. And again, within molecular breeding, you have marker-assisted selection, whole genome prediction, or genomic selection. So what I am discussing today is basically this particular aspect, the molecular breeding aspect, and how it is impacting the seed product development in a big manner. And one of the biggest advantages of this particular discipline is that it is free from regulatory concerns, right? So as compared to genetic engineering or genome editing, where the products are, no doubt, very impactful products, but then they have to go through certain regulatory approvals in many different countries, right? Whereas in this case, this is free from all regulatory concerns. The products are as good as the plant breeding products. So, and just to give you some context, when we had these RFLPs, the evolution of molecular marker technology, the central point of the central message in this slide is that when we were in the RFLP and the SSR world, we were generating genotypic data points in thousands. Whereas on the advent of SNPs, we are now generating marker data points in millions, and in fact, in billions of data points, right? So one of the biggest challenges right now is how do we make sense of this voluminous data into meaningful insights that breeders can act upon. So that is a bigger challenge than generating the genotypic data point itself. And one of the biggest impact this has had, high density genotyping, is that it has dramatically reduced the cost of genotyping per data point. So that is the reason why breeding programs are considering using more and more genotypic data points for making selections. So how is all this being used? So you are generating millions and millions of genotypic data points on one hand, and then just Karen talked about the precision phenotyping approaches, right? So precision phenotypic data points are coming from another side. So how do we number crunch all these things using statistics as well as some computational infrastructure? So all of them get utilized in 2 major streams. One, you map a particular genomic region responsible for a trait, and then you select using the markers for that particular trait. But this is very, very effective, but then this is not applicable in all the contexts. Say, for instance, grain yield, which is highly quantitative. There are multiple genes with small effects. It's literally impossible to identify each and every gene and go after them. So there is an approach called whole genome approach, where you genotype the entire genome and then you make selections or predictions based on that whole genome approach, which is called whole genome prediction or genomic selection, one and the same. So now just to give you some context with respect to the single gene or fewer gene approaches, these are all very well-known diseases in rice, a very popular crop in this part of the world. So be it bacterial leaf blight or rice blast or brown plant hoppers, there are major genes influencing these traits. And they are all thanks to molecular markers. They have all been mapped, cloned, and very good markers have been developed, which are very, very efficient in selection. But unfortunately, the same approach cannot be said or cannot be used for traits like grain yield, which is, as I told earlier, highly quantitative and there are thousands of genes acting in unison to give you the granule. So the breeders responded brilliantly to this particular context where they were not able to use markers associated with specific genes. So then they came out with this concept. It was originally the animal breeders that came up with this concept that you have a large population of individuals. So can I select a smaller subset of the individuals which are representative of the larger population? And I study them, study their genotypes and study their phenotypes in the field. And I come up with a model for estimating the marker effects. And then I use that model to predict the possible phenotype of rest of the individuals by only genotyping them. And I don't phenotype them anymore because phenotyping is much more expensive nowadays in the field. And then you evaluate in silico virtually, and then you rank all those individuals, and then you cut out the lower and the average ones, and then you select the top 1% or 2% or 3%, whatever the breeder is intending to do. And this particular approach is actually influencing each and every stage of a hybrid breeding program. Be it at the beginning where you make crosses, or when you are advancing the progenies and coding them as possible inbreds, or when you are making hybrid combinations using those inbreds. This particular approach is being heavily leveraged now in all these stages of the breeding program. Just to give you one example, say for instance, if you're a corn breeder, you would know. So you have a heterotic group A and heterotic group B. Let's say you have 100 inbreds on A, 100 inbreds in B group. How many possible combinations possible, right? So you can make 100 by 99 divided by 2. So those many combinations are possible. And it's literally difficult to make all possible combinations in the field and test them because it's very expensive. Whereas this particular technology, whole genome prediction, even if you have 100,000 inbreds in one particular heterotic group, you can make all possible combinations between those groups and in silico, and then you evaluate them virtually, and then you make only the top superior combinations in the field. So this is something unprecedented. It was unimaginable just 10 years ago, but now this is a reality, and many of the leading breeding programs do this. And this is not any more theory, and there are very successful stories to tell. So within Corteva, we came up with a product called Aqua Max, which is water efficient corn that was very popular in North America very recently. It was designed to produce more crop per drop. It had very efficient root system, better stay green traits, displayed tremendous reproductive resilience. And this was a product of whole genome prediction, managed stress screening, as well as precision phenotyping that Karen talked about. So I think this is how all the technologies are converging to give you products. And this is not any more theory. These are impacting the product development in a big way. And similarly, we are using the same technology for coming up with a number of high quality, grain quality rice hybrids in the Asia Pacific region. So, I mean, one thing that I get questions on this, if it is so popular, why are not people doing this very successful, right? Across public sector programs, across many of the private sector programs, many have tried, they've not been very successful. So I think one of the important reminders here is for the breeding, for this kind of approach to be successful, this doesn't act in isolation. Unlike the specific marker-based approach, where you can just identify markers and use it in any breeding program, that works very well. But the whole genome prediction program, the approach, it will work well only when there are certain enabling elements. So for instance, one, you should have your germplasm very well organized into heterotic groups. So for instance, in Corteva, so what you see is a universe of inbreds. Now, if it is like this, it's very difficult to make use of whole genome prediction approach. But if you can find a way to organize your breeding material into defined heterotic groups, and if you follow all the breeding disciplines of making crosses within heterotic groups and then making hybrids across heterotic groups, all those breeding disciplines, if they are strictly followed, and if you keep your germplasm very well organized, then the whole genome prediction is going to be much more effective. So without good documentation of breeding history, the strategy is not likely to be very useful or very successful. Similarly, another important point is, in any hybrid crop, you need good inbreds, which are parents of the hybrids, right? And breeding programs invest heavily in inbred development. And these inbreds need to be developed in large numbers to be able to apply whole genome prediction approach. If you produce only a handful of inbreds, then the whole genome prediction is not going to be so useful. So for instance, in corn, doubled haploids has become already very popular. So 95% of the breeding programs globally use double haploids, and it is possible to produce literally millions of homozygous lines each year across the globe. Similarly, rapid generation advancement of speed breeding in rice. So we are able to generate large number of inbred lines each year, year after year, using these technologies. And this is one technology that is actually making the whole genome prediction successful. And the most important part is when you have large number of genotypes, phenotypes. So the important part is being able to handle all that information very efficiently, which is the breeding informatics component, right? So being able to crunch all that data and come up with easy visualizations that breeders can really act upon. So that's another important element. This one. So how all this is actually impacting the plant breeding. So if you see, I'm sure you might have heard about this breeding funnel, right? The plant breeders used to put lots of material on the top of the funnel, and then it comes through the filter and ends up with a handful of commercial material. So the longer the funnel means the product is taking more time to be developed to the reach to the commercial stage. So in the past, the funnel used to be very long, whereas now with all the the speed breeding approaches and the whole genome prediction approaches, the funnel has been considerably shortened. So which means you are able to develop products much quickly. And also the another important impact on the funnel is that the funnel is broadened considerably, in the sense you are able to produce large number of recombinants and you are able to pour all those recombinants into the funnel. Whereas earlier, we were able to generate only hundreds of inbred lines in a year. So you were able to put only a fewer number of recombinants in the funnel. Whereas now it is quite broad, quite short, and it has improved the entire breeding process efficiency. And I'll just end with this particular slide. So I'm sure you all must be knowing Rex Bernardo, a very famous corn breeder from University of Minnesota. So once he was asked by one of his students, can you summarize plant breeding in one sentence? So this is what he said. So he said, select the best, cross the best, and then you hope for the best. So I mean, in simple terms, this is what plant breeding is, right? So this used to be the paradigm in the past, but now what's happening is with the advent of all the technology, be it on the genomic side or on the inbred line development. So now it is slightly changing in the sense, you are still selecting the best, you are still crossing the best, but then you are not leaving the rest of the process to chance. So now you are able to create that outstanding genotype that you have in your mind with a lot more precision. So you have better control over the process and you have better control over the end product. So, I mean, this is success by design and not anymore success by default. Right. So I think that's how the technology has impacted the breeding program in a very positive manner. And we look forward to having, much more technological advances in this field. Thank you very much for the opportunity.
Thank you, Babu. Shivam, are we able to have Dr. Rajam online?
Yeah,
Yes.
I am there.
Okay.
I'm already on the online. Yeah.
Okay. So, Dr. Rajam, can we go ahead with your slide deck?
Yeah, sure. Yeah. Yeah, can you see the slide?
Yes, Dr.
Hello?
Rajam, yes. Yeah, we can see.
Okay, yeah. Okay. Sorry for the internet problem. Firstly, I would like to thank Professor Ramanathan and all other organizers for giving me this opportunity. Let me start with a very brief introduction. As you all know, all crops are affected by a variety of pathogens and pests. which cause a colossal loss of crop yield as well as quality. So therefore, crop protection is absolutely important for enhancing the crop yields using conventional and non-conventional methods. So the conventional methods like use of agrochemicals or uh, and other like non-conventional method like planting technology, each one of these methods have their own limitation. And recently, RNA interference has turned out to be a novel and potential alternative for crop protection against pathogens and insect pests as well as nematode parasites. Besides the improvement of many other agronomically important characters like delayed ripening or induction of motility, etc. So today, I shall be giving an overview about RNA interference in crop protection. So basically, RNA interference is a gene silencing technology which has tremendous, applications, potential applications in fundamental research like for deciphering gene function and applications, a variety of applications in agriculture, not only in agriculture, but also in industry and the medical field. In agriculture, you can develop crop plants with resistance against pathogens, pests, and improvement of many, many other agronomically important characters. So the RNA interference phenomena was discovered by these two scientists, Andrew Fire and Craig Mello from US. And they have demonstrated that RNA, the double-stranded RNA is a triggering mechanism for RNA interference in the host organism, which can lead to the regulation of gene expression. And this groundbreaking work was published in Nature in 1998, as you can see here. So the RNA interference is basically sequence-specific gene silencing, which is mediated by presence of double-stranded RNA, right? And now we know that RNA interference is a very ancient innate immunity mechanism, defense mechanism against viruses in plants, animals, and humans. Like when a plant is infected with a virus, the host plant make sRNAs, small interfering RNAs, against all viral genes. That is how the viral multiplication, replication is blocked because of sRNAs which are produced by the host plant. And we also know that RNA pathway is highly conserved across eukaryotic organisms, and which is, RNA is involved not only in the regulation of gene expression, but also in the chromatin remodeling, inactivation of genetic elements, transposable genetic elements, besides, their involvement in the regulation of growth and development, signaling mechanism, and also response to stresses, both biotic and abiotic stresses. Yeah. Yeah. So as I said, double-stranded RNA can trigger the RNA pathway, and how the double-stranded RNAs are formed in the eukaryotic cell. It can be formed by a variety of ways, like viral infections or introduced foreign genes, transgenes, or hairpin RNAs, that is aberrant messenger RNAs with secondary structures, read-through transcription of multiple genes, pseudogenes, transcription at the repeat sites, so repetitive elements. All this can lead to formation of double-stranded RNA. And once the double-stranded RNA is formed in the cell, it can be readily cleaved by RNase 3 enzyme called dicer into small interfering RNAs of 21 nucleotide size. And this step is called dicing, right? Formation of sRNA molecules, sRNA duplex. And now the sRNA molecules can be recruited in a multi-protein complex like RISC, RNA-induced silencing complex, where the arginate protein, which also belongs to RNase 3 enzyme family, is a part of this multi-protein complex. So now, in the RISC complex, the sRNA duplex, it has both passenger and guide strand. Passenger is also known as sense strand, and guide strand is also known as antisense strand. So interestingly, the passenger strand is selectively degraded in the RISC complex by the action of AGO or arginate protein, And selectively, the guide strand retains with the RISC complex. Now, the activated RISC complex along with the guide strand of siRNA can not only identify the cognate messenger RNA or target messenger RNA, this complex can also degrade or cleave the target messenger RNA. And as a result, there won't be any protein. So that means the gene silencing is occurring after the transcription. So therefore, this pathway is also known as post-transcription gene silencing, or PTGS. And for silencing any target gene, we generally go for hairpin RNA construct, where we clone the partial sequence of the target gene in sense orientation and as well as antisense orientation with a spacer, a very small size spacer DNA, which is usually a small functional intron under the control of an appropriate promoter. So this is a typical RNA, hairpin RNA construct, which can be deployed into plants for the silencing of a target gene either in the host plant or in the pathogen or pest. Yeah, so here I'm showing the basic concept of RNAi for crop protection. And this is popularly known as host-induced gene silencing, or HIGS, which is also known as host-delivered RNA interference, right, or host-induced RNA interference. But I would love to call it as a host-induced gene silencing, or HIGS. So first and foremost important is the identification of a potential target gene, either in the pathogen or insect. For example, in case of insects, we have identified quite a few novel targets for RNAi. I'm just giving one example, that is chitin synthase. As you know, chitin synthase is involved in the synthesis of chitin molecules, which are a very integral part of the cell wall of pathogen, fungal pathogen, as well as insects. And in case of insects, of course, chitin molecules are involved in insect metamorphosis. So if you knock down expression of chitin synthase gene by RNAi, so obviously it will lead to insect mortality, because
Dr. Rajan, just a second.
Yeah, sure.
Yeah, so if you can actually focus more on the application part, then we will have
and I'm coming to that
for the questions.
For the sake of I'll do that. For the sake of audience, of course, I've given a brief introduction. So the selected partial sequence of the target gene can be used for the preparation of hairpin RNA construct, which can be used for the plant transmission. Now we expect that transformed plants will produce double-strand RNA and siRNAs against the target gene of the insect. And when the insect larvae eats on the plant pot, after the digestion of the plant pot in the insect gut, the double-strand RNA and siRNAs are available, and these molecules, small RNA molecules, can utilize the insect RNA machinery to target its own gene, chitin synthase gene of the insect. So as a result, the final outcome would be pest-resistant plants. So this is the very basic strategy which is being used for not only for the development of insect-resistant plants, but also the resistant plants against viruses, fungal pathogens, even bacteria and nematodes and mites. Yeah, I'm coming to the practical application. Like in my lab, we have developed tomato plants resistant to a very notorious insect pest, Helicobacter parmigiana, by RNA silencing of a target gene, that is chitinase. And we found, after silencing of the target gene, chitinase gene in the insect, we found that there was about 60% larval mortality, which is due to a significant dramatic reduction in the transcripts of the target gene chitinase. And this has also led to decrease in the larval weight length and decrease in pupal weight. And also there was a developmental time delay by about a week. And also we also found that there were deformities at pupa stage as well as adults. And more importantly, the insect fecundicity was also affected. Egg laying, egg hatching was also affected. This work was published in Plant Molecular Biology in 2016. And we have also developed the transient tomato for fungal resistance against Fusarium wilt caused by Fusarium oxysporum by knocking down of ornithine decarboxylase gene of the fungal pathogen. And as you can see here, these are the control, non-transgenic control plants which are showing typical wilt symptoms after challenging with the Fusarium spores. but the different RNA lines, they have shown a very high resistance to moderate, because of expression of siRNAs against the fungal gene, that is ODC. Yeah, so very recently, there's a very innovative approach, that is GMO, genetically modified organism free RNA approach, which is popularly known as environmental RNAi or eRNAi, and where we spray double-strand RNA externally onto the leaves or onto the trunk, and this is also popularly known as spray inducible gene silencing 6 as against the Higgs, which I discussed just now. And here, and even we regard this, the use of double-strand RNA as a pesticide, a new generation of crop protection against the pathogens, pests, and when the double-strand RNA delivery can be done by directly, but double-strand RNA can be easily undergo degradation by cellular nucleases. So therefore, people have used the nanoparticles like biopolymers, for double-strand RNA delivery, or carrier proteins, peptides were also used. And very interestingly, E. coli, a particular strain of E. coli called HT115, expressing double-strand RNA against the target gene of the insect, can also be sprayed. And this is also a very viable approach. And the double-strand RNA delivery approach has been used for the control of insect pests, pathogens like viruses, virides, bacteria, and fungi, and also the parasitic nematodes and mites. The problem is, of course, this has a very short period of protection window, only 5 to 10 days against viruses, etc., because of double-strand RNA eventually degrades. So therefore, you have to periodically spray the double-strand RNA for continuous protection of the plant against pathogens and pests. The exogenous delivery of double-strand RNA can be done by different ways. It can be sprayed onto the leaves, or foliar spray, or seed coat, or trunk injection, or drop irrigation. All these methods can be used, but I think in most cases, the double-strand RNA was very effective or efficient when you spray onto the leaves. And siRNAs, the spray with not only the double-strand RNA, with also siRNAs is also very effective. The long-distance spreading of stakes associated with double-strand RNA or siRNAs, suggests that uptake of spray double-strand RNA by the plant is via stomata, and we can also see systemic translocation of siRNA or unprocessed double-strand RNA via the vascular system, that is phloem or xylem. I'm almost through. Another thing is the quantity. How much quantity of double-strand RNA should be required for the field scale control of pests and pathogens? And it has been suggested that metric ton levels of double-strand RNA would be required for the field scale management of pests and pathogens. And it was roughly estimated that 10 grams of double-strand RNA per hectare is required, which is a huge quantity. So therefore, the large scale synthesis of double-strand RNA with the low cost is required. And the following methods can be used for the preparation of double-strand RNA: chemical synthesis, in vitro transcription, E. coli expressing double-strand RNA. Among these three, in my opinion, the last one, E. coli expressing double-strand RNA, is most viable approach for large-scale synthesis of double-strand RNA. And if you make a comparison between transmittive, the genetic transmission, or non-transmittive, the development time and development.
Dr. Rajam, sorry for interrupting.
I have only a couple of slides, but I'm almost through. So if you make a comparison, non-transmittive has quite a few advantages over transmittive, like less developmental time, developmental cost, etc., and less of regulatory process, or acceptance by consumers is very high with non-transmittive. I'm just giving one example of successful example of double-strand RNA spray, like 6-mediated control of Fusarium graminearium on leaves sprayed with CYP3 double-strand RNA in wheat. You can see a very, very good control of this fungal disease by double-strand RNA spray against the CYP3 gene, target gene, as compared to mock control and GAP control. So there are many other successful examples of control of insect pests and other pathogens. So eRNA advances, of course, now it has been beyond doubt, it has been proved that the exogenous application of double-strand RNA molecules, either siRNAs or double-strand RNAs, can protect crop protection against pathogens and pests by triggering RNAi in a GMO-free manner. So therefore, the exogenous application of RNA in plants has efficient in triggering RNA of the plant genes against viruses, viruses, etc. And the conjugated RNA molecules through nanoparticles or carrier peptides greatly enhances their resistance to nucleases and efficient delivery. And as I said, the sRNAs can also trigger RNAi and effectively control pathogens in pests. Yeah. So more recently, people are also using artificial microRNA technology. The artificial microRNAs are nothing but, we use a precursor microRNA where we replace this microRNA duplex region with the sequence of our interest against a pest gene or pathogen. Now it has become an artificial microRNA, and this can be processed in exactly similar manner like native microRNA to produce a mature microRNA, which is involved in the silencing of a gene. And we have used very successfully, we have used artificial microRNA technology to knock down expression of ecdysone receptor in Helicoverpa for developing insect-resistant tomato plants. Yeah, so we could see a very significant mortality, about 70–80%, and drastic effects on insect growth and development. All this is due to more than 90% knockdown of the target ecdysone receptor gene. Even the knockdown of the downstream genes to ecdysone receptor were also reduced. The transcription levels were also reduced. Yeah. So as you know, the gene editing, I'm not getting to details. Then a very
Dr. Rajam, I think
last slide, please. Yeah, last slide. There is a very excitement about gene editing, the CRISPR-Cas system for gene knockout for the crop improvement program. I'm not going to discuss this in the, And finally, I would like to say that RNA is a blue moon of biotechnology or technology for the 21st century because of many advantages of RNAi, double-strand RNA, even artificial microRNA, over the conventional transgenic approaches for crop protection against pathogens and pests. Thank you.
Thanks a lot.
I've extended. I'm sorry, I've extended a little bit. Yeah.
Thank you. So Shivam, when we are waiting for the questions from the audience to come, what I thought was I will just quickly try to integrate the highlights from all the eminent panelists we have had today. We started with Nimrod, who actually explained the promise behind no spray technologies. No spray technologies, as well as the importance of ensuring that farmers will have access to the novel technologies that are being developed, which is a key component, because without that, the technology developed has no reach and bears no meaning in terms of large-scale deployment, especially when it comes to small-scale farmers or smallholder farmers. Karen touched upon the challenges in phenotyping plant physiological parameters, and she also expressed the elegance of the plant array system that has been developed by Biotech to study in real time continuously the physiological parameters, which can greatly enhance the speed at which we can understand the parameters that are impacting key attributes, and maybe we can integrate the areas touched upon by Babu in terms of using the molecular markers for relating the physiological insights we are getting to the genotypic information from the plants we are studying, and quickly arrive at the genetic basis of the key attributes we are chasing so that they can be more effectively deployed, especially for the traits that are, I would say, let us say, monogenic or oligogenic, or use the whole genome prediction approach or the genomic selection approach if it is, let us say, a lot of loci with small effects, like Babu explained for yield. And finally, Dr. Rajam elegantly explained the concept of RNAi, which can be through a GMO approach in plants, or can also be through a topical application. And the topical application especially is a very, very promising approach. But unfortunately, like Nimrod was highlighting, the regulatory framework for bringing such products to market is painstakingly slow. So I would urge all the people who are participating in forums like this, they may be speakers, panelists, or audience, should actually contribute in their own ways to ensure that the message reaches the policymakers as well as the regulators, and we create an environment in which the speed with which technologies are being developed also leads to speedy deployment of technologies using models that make these technologies affordable to smallholder farmers. I hope actually I was able to combine the themes that all the panelists brought together. And in the meantime, Shivam, I wanted to check with you if we have any questions, or we have time for questions from audience. Shivam? Shivam, can you hear me?
Yes, now sir, I can hear you.
Yeah. Do we have time for taking questions from audience?
Yes, sir, we do have time for the questions, right?
Okay.
Actually, this is the last session, so we can overshoot, right? No issues with that.
Oh, okay. In that case, actually, I will take a few more minutes and then maybe prompt some of the panelists to delve a little deeper and throw more insights on some of the key concepts they touched upon. So my first question will be to Nimrod, to maybe summarize the disruptive business model he is talking about, with which the mango export potential of Senegal was doubled in just a year. Nimrod?
Yes.
Yeah.
Well, we used an Israeli model that was very common at the first 60 years of Israel, and we applied to emerging markets and how this is working. Today, when a company, a technological company, is going to the farmer, it is suggesting the farmer come and buy our product or our service. So this is one way. What Green Valley is doing, it is saying to the farmer, we go together. We will help you to improve the produce that you are growing, the quality and the quantity, and we will help you to get it to the market. Now, if all of us are going to do better, then we will get better price, and you will get more income, we will get more income. This is a value chain approach. It is not just one product. One product in the case of mango is critical, and we couldn't do without this, which is for fruit flies. But other than this, if you think of a farmer in India that today the yield is about 3-4 tons per acre, while in Israel it is about 10 times more, 10 times more. So you start by dealing with critical problems, but then you need to do other things. Now, using this model, when we have agreement with the farmer, we can have on board also other experts, other companies, other technologies. Now we have a business model that we can pay them, because let's say irrigation, using irrigation, now the farmer is having more yield. So now we can pay the company that is selling irrigation. Without this model, the company can have no access to the farmer because the farmer cannot finance it in advance. Okay, so this is a business model. It is a platform, just like if you want, you can think of Amazon that is connecting ants. This is connecting on a value chain. And as long as you add value, real value, which translates to market, to selling more yield, higher quality for more money per hectare, you can get in and you can make money on this. And it's all about making money.
Thanks, Nimrod. I'll come back to you. Karen actually has another appointment. I'll just quickly touch upon one question that I had. Karen, you said the timeline for phenotyping can be dramatically reduced. But see, there are special situations where reproductive stage drought, let us say in a crop like maize, the reproductive stage drought has a very different kind of impact in comparison to the early stage drought, provided there is water available after that stress. So for addressing such things, do you have any other accelerated approach rather than waiting for the gradual plant growth to get to that point?
So what we see, and this is after a long experience, over 10 years now, with using the system, is that the plants, as much as they gather more biomass, grow faster and better in the first stage, they will then provide more yield at the latter stage. So even though if there are additional processes at the latter stage when it's flowering and so forth, still, whatever the plant succeeded to gain in the first vegetative stage, he will use that. This is like his resources for a guarantee of success at the further stages later on. So if we do see in the vegetative stage that there is a great transpiration levels, a great rate of transpirations, of biomass growth, this specific plant versus other plants that are measured at the same time, we can conclude that this specific plant will have better chances to get to higher yields in the field. And we do see it when we compare the greenhouse measurements to field results. We do see very high correlation, about 94–95% correlation to the field, which is very high correlation. Of course, it will never be one-on-one because this is still in a greenhouse and not in the field, but 95%, 94%, it's very high. And when there is stress applied, it's still above 80%. So it's really high correlation. We're going to have a paper published on this element soon with a very thorough analysis demonstrating that.
So Karen, to summarize, you are in short saying that by ensuring source, volume, and quality, you can assure sufficient assimilates or carried over to the sink and provide the yield, irrespective of whether there is a later stage stress or not.
There could be some influence later on, but it will not be too major to change the results. Yes.
Excellent. Thanks a lot, Karen. And,
sure.
and feel free to drop off whenever you have to. And in the meantime, I'll probably move to Babu.
I would just, if I may, I want to thank you all for inviting me and also participating with this great forum. And it was a great pleasure. I also learned quite some for the presentation. So I thank you all and and success.
Thank you, Karen, for your time and your insights. Thank you. And wish Biotech the best.
Thank you.
Thank you, Karen.
So Babu, you actually highlighted the importance of having structured germplasm in the breeding program to assure greater value capture using genomic selection, right? So does it mean in this era where we will have to basically accelerate everything, even if a company is getting started today? In that kind of a scenario, what do you suggest should be the approach for companies that don't have a long breeding history to get to that point quickly?
Yeah, so, well, I think some amount of data is something that we can't do away with. But for instance, many of the public sector breeding programs, I have been part of the public sector breeding programs earlier. So I discussed with my professors in IRI and other reputed institutions. We discuss this very often. So how can those breeding programs they have accumulated a lot of data. Say, for instance, the simple AICRP trials, the All India Coordinated Project trials. Tremendous amount of information is already there, right? But then that is not being put to use except for selecting for that particular year or maybe for the last 3 years, right? And then it is not being used. Whereas what enables the genomic prediction is a good documentation for the last, let's say, 10 or 15 years of that data. And if that is there, I think that becomes a very good foundation. Similarly, for any new breeding program, new company that is trying to do, wherever they are acquiring germplasm from the, they must be acquiring it from another company or from anywhere. They will have to spend at least some amount of time, say at least for the next 2 or 3 years, good amount of phenotyping. I mean, we can't do away with phenotyping completely, but some amount of energy and time and investment will be needed in the beginning, at least for 2 or 3 years. But once a certain minimum critical threshold of data points are developed, that can be, again and again used.
So, Babu, to summarize, I may be oversimplifying, but just to summarize for the benefit of most audience, in short, you're saying if you do not have history, borrow materials that have history and get access to all the background information.
Absolutely, yeah. In simple words, yes.
Right. Thank you, Babu, for that advice. Dr. Rajam, you actually touched upon the immense potential the sprayable RNAi technologies can bring, obviously because of the GMO related limitations that will pop up in many countries. What do you think we should be doing in order to ensure that we create the appropriate regulatory environment for the sprayable RNAi solutions? Dr. Rajam?
Yeah. Okay, can you hear me?
Yes.
Am I audible? Yeah,
yes.
okay.
Yeah.
The sprayable double-strand RNA or siRNA is a very innovative approach. This has only one limitation, right, where the regulatory process may come in. The long double-strand RNA or siRNA, if they have sequence similarity with other genes, or genes of beneficial microbes in the soil, right, or beneficial insects, etc. So they may, we may also see silencing of unintended genes, which may lead to some effects on other organisms. This is the only concern. The other concern is otherwise, the regulatory process may be minimal with the double-strand RNA.
So, Dr. Rajam, for the first part you mentioned, it should be easy to identify
The silencing
targets.
of genes.
Yeah, for the first.
In general.
Yeah, please go ahead, Dr. Rajam.
Yeah. So, in general, there is a very excitement about double-strand RNA spray because it's a very easy, innovative approach, right? The other concern is we need a huge quantity of double-strand RNA. That can be probably overcome by using E. coli expressing double-strand RNA, because E. coli can be grown in big fermenters. So so that you can have huge amount of double-strand RNA. But as compared to the RNA transgenic approaches versus double-strand environmental RNA, obviously the environmental RNAi or double-strand spray has an edge over transgenic RNA approach because of its minimal regulatory process.
Yeah, plus, actually,
and
the
of course,
speed
identification
with which,
of target gene may not be a problem because we've now. Yeah.
Now, what I was trying to say is the sprayable RNAi
it takes less time. It takes a very, very less time as compared to the RNA transgenic approach, which takes a hell of time. 6 to 7 years at least. Yeah. And
so once a target is
RNA,
identified,
it won't take much time.
Yeah, the molecules.
That is the added advantage. Yeah. It can be readily used. Yeah.
Right.
That is the advantage of double-strand RNA spray.
Thank you, Dr. Rajam.
Yeah. And
Nimrod,
identification, yeah, I was telling about identification of target gene. That may not be a problem because of availability of complete genome sequencing projects in insects, pathogen. So that may
Right.
not be a problem. Yeah.
Right.
Yeah. Thank
right.
you.
Thank you, Dr. Radhan. Nimrod,
Yeah.
actually, you were kind of suggesting a business model where value capture is done after ensuring value is created. Is my understanding right?
Can you repeat it, the question?
No, what I was saying is from the way you were explaining the business model, I kind of came to the simplistic understanding that in that business model, you are thinking about value capture only after value is actually created at the hands of the farmer.
Not necessarily. The farmers can and should participate partially in the payment in advance. In some cases, even the government. But in this case, if the company is not sure about the quality, about the added value that it is really giving to the farmer, they will get nothing. So, and you know, many are just selling to the farmers and for many years and nothing is changing, which is very much the case in India. If you go to Indian farmers, I know mango growers 20 years ago, they would make more money than today, more money. So how is it possible? Now, if you want to sell, go ahead, sell it, but you can make more money if you get on this business model. And later, if the farmer is making more money, you can also make more money. And
that is exactly what I meant. Yeah.
Yes.
That is
definitely.
exactly what I meant, Nimrod.
Definitely.
And Nimrod, will you be able to share in simple terms what exactly is the technology you are deploying for fruit fly control in mangoes? Is it using a color or light trap and then also capture and kill kind of approaches? What exactly is happening there?
Okay. So it is all about smells. And there is nothing more powerful in the kingdom of animals than smells. Smell manipulate not us less, but more insects better than anything else. But the problem with smell is that we don't know, or we didn't know until recently, how to control it. And if you think about irrigation, drip irrigation, they didn't invent the water. They didn't invent the pipe, the water pipe. They only invented how to control the release of the water. This is what they do. So what BioFeed did is really inventing how to control the release of smell, also developing some special smells. Now we put it on the tree. It's like 30 by 30 centimeters, even less, small post, yellow. But the yellow is not the main thing. The main thing is about the smell, and the smell is released every day as very, very, very little, and during the entire season. So you don't need to service it at all, zero service, and you just attract it. And when it is so powerful, you need so few units. That's it.
Thanks a lot, Nimrod, for that simple explanation of how the technology works. I thank all the panelists for taking time to participate and share their insights to the audience in this session. Thanks a lot, everyone. I thank CII for bringing all of us together in an effort to bring towards greater understanding and also identify potential technologies which can be deployed in a meaningful manner to the farmers in India. Thanks a lot, everyone.
Thank you very much.
Thank you. Thank you.
Thank you, everyone, for joining us today. With this, we come to an end of day 2 of the global dialogue. I take this opportunity to thank the chair and all our speakers for this very interesting and thought-provoking session. We are sure the participants were intrigued with the innovations and have gained from the rich presentations and conversations. I would also like to thank ITC, PI Industries, and New Holland Tractors, our principal partners, Corteva, Rallis, Spartcom, and ADM Agro, our principal co-partners, and NetFM, our associate partner. I would like to inform the delegates that for closing the global conference, we have the last special plenary session tomorrow, starting 7 p.m. I request all of you to join us for the session tomorrow. Thank you once again for joining us today.